Assalamualaikum and have a good day everyone. In this video, we will learn Subtopic 1.2 Meristematic Tissue and Growth from Chapter 1 Form 5. What is meristematic tissue? Meristematic tissue are undifferentiated living tissue in plant which are responsible for plant growth. Meristematic tissues are divided into two types, which are apical meristem located at the tips of plant shoot and root, and the second one is lateral meristem, consists of vascular cambium and cord cambium. This is the position of shoot apical meristem, root apical meristem, and lateral meristem. And this is the location or position vascular cambium and cord cambium. Okay, now let's discuss more about growth process. Zone of cell growth divided into three: zone of cell division, zone of cell elongation, zone of cell differentiation. So this is the picture of apical meristem of shoot. And then this is the picture of apical meristem of root. And then the location of zone of cell division at the shoot. And then the location of zone of cell elongation. And the zone of cell differentiation at the shoot. This is the location of zone of cell division at the roots, zone of cell elongation at the root, and zone of cell differentiation of the roots. So now we will discuss about the activity in each zone. What happened during zone of cell division? Based on their name, so the zone of cell division take place at the apical meristem, which consists of actively dividing meristem cell through mitosis. The increase of the number of the cell causes the elongation of the plant stem. When new cells are forming, the cell formed previously are pushed to the zone of cell elongation. So this is the picture of cell in the zone of cell division. It has small vacuole and very large nucleus. Okay, we move to the zone of cell elongation. What happened during cell elongation? The zone of cell elongation consists of cells that are increasing in size. The increase in size happens through the water diffusion by osmosis and the absorption of nutrients into the cell and stored in the vacuole. Small vacuole fuse to form large vacuole. This process is known as vacuolation. The diffused water exerts pressure against the cell wall, which push, elongates, and widens the cell. This is the process of vacuolation. Small vacuole will enlarge and the small vacuole will combine to form large central vacuole. What happened during cell differentiation zone? The zone of cell differentiation consists of differentiating cells that differentiate once they have reached their maximum size. The cell differentiate to form permanent tissue such as epidermis, cortex, xylem, and phloem. The cells change their shape and structure to become specialized cells and with specific function. For example, epidermal cell in leaf differentiate and form gut cell to control the opening of stomata. And other than that, epidermal cell in root differentiate and form root hair cell. So let's discuss more about the growth, the primary growth. Primary growth is the process of the growth that occur after germination, and it takes place in all plants to elongate their stem and roots. This growth takes place in the apical meristem at the shoot tip and root tips. So this is the apical meristem of the shoot and the apical meristem of the roots. Primary growth start when the meristem cell in the zone of cell division of apical meristem are actively dividing. And then, after that, this is followed by elongation and differentiation of cell. At the shoot tip, leaf primordia and shoot primordia will grow to form new leaves and shoots. This enables the plant to increase in height. At the root tips, the root cap will be adjusted when they penetrate the soil. This causes the cell of the root cap to be replaced by the meristematic cell. So, the primary growth is function to increase in height. Next, we discuss about the secondary growth. The other types of growth is secondary growth. Secondary growth occur mainly in eudicots and a small number of monocots, like shrub, to increase the circumference or diameter of the plant stem and root. For non-woody plants, such as hibiscus plant, secondary growth does not happen. Secondary growth results from the division of lateral meristem located in the stem and root. What is lateral meristem? Lateral meristem is divided into two. First, vascular cambium, located between the phloem and xylem tissue in the vascular bundle. So, this is the location of vascular cambium. The second one is cork cambium, located under the epidermal layer. So, this is the location of cork cambium. So, this is the picture of secondary growth process at the stem. Okay, after 3 years, it will increase the circumference or diameter of the stem. The stem become thickened. So let's discuss more about the secondary growth process at the stem. Okay, this is the diagram of cross section of stem after primary growth. After primary growth, the stem have primary xylem and primary phloem. Secondary growth start when the vascular cambium divide actively through mitosis. And then, this is the location of vascular cambium. Okay, after that, the cell in the cambium ring divide inwards to form new xylem and divide outwards to form new phloem. The new xylem tissue becomes secondary xylem and new phloem tissue become secondary phloem. So, after the mitosis of vascular cambium, okay, the phloem have secondary phloem, xylem have the secondary xylem. Next, after that, when undergoing secondary growth, primary xylem is pushed towards the pith, whereas primary phloem is pushed toward the epidermis. As a result, the primary xylem tissue are compressed to form stronger wood layer. This is because the wall of xylem are thickened with lignin, which is wood. The thickening of lignin provide mechanical support to the plant. After that, when the secondary xylem layer is compressed, there is an addition to circumference of the stem, which cause the epidermis of the stem to stretch and crack. The cork cambium actively divide to form cork cell on the outer side and then cortex on the inner side. The cork layer protect the stem from insect and pathogen attack when the epidermis is cracked. So after undergo secondary growth, the plant stem have secondary phloem, secondary xylem, cork, and the cortex layer. Okay, this is the picture of process secondary growth at the stem. We can see the circumference or the diameter of the plant become enlarged. So, let's discuss the secondary growth at the root. The root of the plant also undergo secondary growth in order to increase the circumference of the root. The secondary growth process at the root is the same as secondary growth at the eudicote stem. So this is the picture of the secondary growth at the root. Started from vascular cambium cell, divide actively and combine to form complete ring. The cell in the cambium ring divide inwards to form secondary xylem and divide outwards to form secondary phloem. After that, due to the vascular cambium activity, the root becomes thicker. The cord cambium located under the epidermis divide actively to form cord cell. The cord cell provide protection to the root tissues. So the structure of the roots after secondary growth have the structure of secondary phloem, secondary xylem, and they have the structure of vascular cambium. The secondary xylem, okay, have the structure of like star inside the root. So this is the picture of the stem at the temperate country. Okay, the stem have annual ring. Okay, the age of plant living in temperate climate can be determined based on the annual growth ring. Why the annual growth ring will be formed? Because during spring there is enough water supply and sunlight. The secondary xylem form is bigger and the wall is thinner. Therefore, the xylem tissue formed in this season are brighter in color. But the growth is unsuitable in summer, causing the formation of secondary xylem to be smaller and thicker wall. Therefore, the xylem tissue are formed are darker in color and it will produce annual ring to determine the age of plant living. Okay, secondary growth of monocots. Even though most of the monocots do not undergo the secondary growth, but some do, such as Yersinia species, Aloe species, and Agave species. So we discuss more about the comparison. Okay, first, comparison between the primary growth and secondary growth. Okay, we discuss about the similarity first. Both growth and can increase size of the plant permanently. Both growth occur in woody plant. Both growth involve cell division by mitosis. Okay, what is the differences between the primary growth and secondary growth? Okay, first, the meristematic tissue involved in primary growth is apical meristem. In secondary growth is lateral meristem. The parts of the plant that undergo growth for primary growth occur on stem, roots in younger region. But in secondary growth occur when primary growth has ceased on mature stem and root. The direction of primary growth is longitudinally, but the direction of secondary growth occur radially. The growth effect of primary growth increase the length of the stem and root of the plant, while the growth effect of secondary growth increase the thickness or circumference of the stem and root of the plant. The tissue and structure formed during primary growth are epidermis, cortex, and primary vascular tissue, while during secondary growth, the tissue and structure formed is bulk, periderm, lenticel, and secondary vascular tissue. Presence of woody tissue. Okay, during primary growth, do not have woody tissue, while during secondary growth, have woody tissue. During primary growth, the thickness of bark is thin, while during secondary growth, the thickness of bark is thick. During primary growth, absence of annual ring form, while during secondary growth, presence of annual growth ring. Okay, so The last one, what is the economic important for the plant that have undergone secondary growth? So, the plant that undergo secondary growth have high economic value. Why? Because they can produce timber like Shorea, Meranti and Balanocarpus, which is cengat. Plant that undergo secondary growth have strong and hard woods, which are suitable to be used as structure for houseboat, furniture, fences, door and others. The presence of annual ring make the furniture look attractive and they can made as decorative item. The woods and bark form the plant. Some plants such as Hopia, Merawan and Meranti can produce resin and oil. The substances can be commercialized as varnish, adhesive substance, perfume and medicine. Flowering can be made as decorative plant. And last but not least, commercialized fruits such as mangoes. Mangosteen are product of the plant that have undergone secondary growth and they have able to generate income as well as boost economic growth. So, with that, thank you.